and it's the 13th of August, 2024, and we're going to interview Prasanth. And would you introduce yourself?
I'm Prasanth Prakash Prabhu, and I'm originally from the southern state of India, Kerala, but currently I'm a PhD student with Dr. Hibbett at Clark University, Worcester, Massachusetts, and I'm studying oyster mushrooms.
Okay, and I've known your advisor for many years, David Hibbett, since he was a graduate student, and it's nice to see him having good graduate students doing interesting work. Now you say you're working with Pleurotus, and most people know this as the oyster mushroom, and they would think, well, maybe you're growing it for food. But you're doing something very different with it.
Yes, actually. So I'm more interested in like how fungi is interacted with other organisms. You know, mycorrhizae is well known, one of the ecological interaction. So I was like, what I can do and what is a system that I can manipulate in the lab for the limited time period I have. So that's why I came across this idea of wood decaying fungi trapping nematodes for getting more nutrients, especially nitrogen. Then I focused my thesis on that, focusing on how does nematophagy have evolved in oyster mushroom family.
And so what are you finding?
It's actually interesting when we talk about nematode trapping mushrooms. Most people think about arthropods that produces constructive rings or similar structures. And I tried to do most of my studies based on the things that we know in arthropods, but I found that it's not the case. It's independent evolution of.
And Arthropods is an ascomycete, so it's very distant.
Yes. Yep. And within Pleurotaceae also, that contains Pleurotus and Hohenbuchelia, it have two genera, and both of those genera can trap and consume nematodes. And I think that it is like independently evolved character within the same family also, because they produces different trapping structures and have different trapping mechanisms to trap and consume nematodes.
Okay, so people, I'm sure, have looked at the relationship between Hohenbuchelia and Pleurotus. Are they very distant? Are they sister taxa?
They are sister lineages, but still they were able to produce distinct trapping structures. They don't. Their trapping structures are.
I'm still walking in the.
Their trapping structures are like not similar. They produce a distinct mechanism. For example, in the case of oyster mushrooms, they produce a neurotoxin that can paralyze the nematodes within seconds. Whereas in the case of Hohenbuchelia, they produce adhesive lollipops that can immobilize nematode upon contact. So I think that it's like very interesting and fascinating.
Yeah. So do you know what the toxins are? They're toxins produced, right?
Yes, they are toxins. So there have been numerous studies trying to elucidate the chemical moiety that causes the paralysis in nematode. If we date back a decade ago, it was considered as a fatty acid. Then a recent paper suggested that it could be a volatile ketone, but I still think that it's not a volatile ketone. It could be some other compound that causes the paralysis.
Yeah. Okay, and your latest results are being presented here in a poster?
Yes, I am presenting a poster, but it's not on oyster mushrooms. I am presenting on Hohenbuchelia, and where we know that they trap the nematode to get more nitrogen. So I did a simple choice experiments to see does the fungi prefer a particular nitrogen source or a nematode. And I was able to show that if you give organic nitrogen, you can prevent the nematophagy in Hohenbuchelia.
And so they don't eat nematodes?
No, they don't eat nematodes if you give organic nitrogen.
Really interesting. Do you know if the same thing happens if you do the organic and inorganic nitrogen with Arthropods? Has anybody done that?
So in the case of Arthropods, most people have looked at adding the particular nitrogen source in the system, and mostly they counted the number of traps produced by the organisms, but they haven't actually measured their ability to trap and consume nematodes. So I don't know what will happen in that system.
Okay. That's very interesting. Now, we skipped over much of your life in India. You were born in the south of India.
Yes.
And where did you go to school?
So I had a bachelor's and master's in botany, and when I was doing master's, one of my mycology professor, Dr. Manimohan, he took us to a field trip into the nearby botanical garden. And he introduced me to a fungus called Orchiloscypha anacardiola, and I was like really fascinated by the interaction of that fungi. The reason is that it's a rust. It's like related to the rust fungi, which are like plant pathogens, but this fungi, instead of parasitizing plants, it parasitizes the insects that are parasites of the plant. So like that thing like blew my mind, and I'm like, the fungal interactions are fascinating. And at that time I realized that there is so much to do, and there is so much that we don't know about the fungal interactions. Yeah.
Was your family interested in biology or fungi?
So my father gave me the freedom to choose whatever whatever I wanted to do. And there was interesting because like my father sells peanut in the beach, and one day he gave me, there was a sprout of peanut, and he gave me that to see like to grow and see. And at that time I realized that actually the peanuts are not produced on the top of the plant, they produce as a stalk and like produce the underground. So like indirectly he was like influencing me to like steering me towards the biology, and like the fact that why they are not like produced in the roots but a specialized stalk from the flower.
That's why we call them groundnuts.
Yes, but they are not produced in the roots of the plant.
Right. No, that's interesting then. Yeah. So when did you come to the United States?
So I came to the United States on 2019, September 3. In the fall semester, I joined the program. As you know, it was like 6 months before the pandemic.
Yes.
Yes.
And so you got a lot of research done, or could you go into the lab?
No. So since I was like just joined the lab, there were other people in the lab who had like priority to complete their experiments. So during that time, I was like mostly looking at the literature and trying to figure out what I want to do with my project.
So a lot of library research.
Yes, and reading.
On the computer, maybe.
And learning about new stuff, learning about new tools, languages, and the other stuff.
Oh, very good.
Yeah.
Well, I look forward to reading more, and I hope I'll get to see your poster.
Yeah. Thank you so much.
Okay. Thanks.